Every year, 120,000 people in the Netherlands are diagnosed with cancer, while 45,000 people die from the disease. Cancer is caused by DNA mutations. By identifying these mutations, we can tailor treatments to bring more benefits to the patient. DNA consists of a unique code of 3.2 billion letters. Each position contains one of the following 4 letters: C, G, T, or A. Each letter represents a specific base. A comprehensive DNA test requires a sample of the tumor and a test tube of blood from the patient. These samples are obtained at the hospital and then sent to Hartwig Medical Foundation. To find out exactly what has changed in the DNA of the cancer cells, we compare the DNA of the tumor sample with the DNA of the blood. The blood contains healthy DNA, and by comparing the two, we can identify mutations that may influence the choice of treatment. The analysts get to work as soon as the patient's blood and the tumor tissue are delivered to the laboratory. We also register the samples in our system. First, we extract the DNA from the blood. We then extract the DNA from the tumor tissue using a machine that washes the DNA clean. All the steps are automated as much as possible in the laboratory, both to provide consistent quality and to ensure the attending physician and the patient get the results of the comprehensive DNA test as quickly as possible. We place the tumor tissue in a device that rapidly shakes back and forth and breaks down the cells, releasing the DNA. We then check if we have enough DNA to perform the comprehensive DNA test. The next part of the process is the most critical. In order to read DNA, it first has to be broken into pieces of about 600 DNA letters long. This is because the machine cannot read 3.2 billion positions at the same time. We use sound vibrations to do this. We then add artificial pieces of DNA to the ends so that the machine can read the DNA sequence. These steps have been automated with a self-programmed robot. To decipher the unique DNA code, we use sequencers which read the sequence of these letters. This is your DNA. To this end, we bring the pieces of DNA together in a flow cell, which is a glass plate to which the pieces of DNA become attached. Next, we add chemicals that give the letters a distinct color. The camera in the sequencer can identify these colors and so reads the sequence of your DNA. To do this as accurately as possible, we repeat this 30 times on the samples of DNA from your blood and no less than 90 times on the DNA from the tumor. This is because tumor tissue often does not consist only of tumor cells. Our sequencers need only 2 days to simultaneously analyze the DNA of 36 patients. The sequencers produce a tremendous wealth of data. To give you an idea, if you typed out the data of a single patient on sheets of A4 paper, you would get a pile as high as the Dom Church Tower in Utrecht. Hartwig Medical Foundation's international team of bioinformatics scientists and software developers has been working for years to develop programs and algorithms to analyze this data. These programs are unique in the world and are available for free to other researchers. The sequencers send the data to the software on our servers, which compares DNA from the tumor tissue with DNA from the blood sample to find the differences between them. Not only can letters get mixed up, but also whole pieces of DNA. Sometimes pieces will be missing, and sometimes new pieces will have been added. The algorithm then links the identified DNA mutations to all potentially available treatments, which it retrieves from international medical databases. The results are recorded in a preliminary patient report. Based on this report, a clinical molecular biologist prepares a pathological interpretation and summary for the attending physician and the patient. This includes an assessment of the available treatments for the relevant DNA mutations, including possible clinical studies in the Netherlands that the patient could participate in. This increases the chances of finding the best treatment method for the patient. Hartwig Medical Foundation will send the report to the attending physician within 10 working days of receiving the blood and tissue samples. The information in the report is combined with all other patient data to carefully determine the best treatment plan together with the patient. If you want more information, please visit oncoact.nl.